Pharmacology usually targets the specific molecular pathways of a substance, attempting to block or mimic a drug at its unique receptor site. A 2022 trial for alcohol use disorder showed psilocybin with psychotherapy led to 9.7% heavy drinking days versus 23.6% for the placebo. Acts purely as a serotonin 2A receptor agonist. Psilocybin lacks any direct action on the dopaminergic, GABAergic, or nicotinic reward pathways that sustain these compulsive behaviors. Standard addiction medications, like naltrexone or varenicline, are specifically engineered to compete at those exact molecular receptor sites to block the addictive process. A single compound is producing sustained remission across pharmacologically distinct addictions without touching their respective reward pathways. The therapeutic mechanism operates at a level of organization entirely above the receptor. Conventional psychiatry operates on a specific baseline assumption. Psychological distress and addiction are treated by identifying a localized molecular deficit and adjusting the corresponding receptor. The cross-substance efficacy of psychedelics breaks this molecular reductionist model. The clinical outcomes cannot be explained by the pharmacological inputs alone. Liberation psychiatry offers an alternative best available strategy for meeting genuine needs, like emotional regulation or containment within constrained field conditions. The standard clinical question asks, which drug competes at this specific receptor? The field-based approach asks, what need is this meeting, and how do we expand the psychological field to meet it differently? By focusing on the pharmacological molecule rather than the broader contextual field, the psychiatric industry is optimizing the wrong variable. The neuroscientific mechanics of this process are best described by the Rebus model, developed by Robin Carhart-Harris and Carl Friston. The brain operates as a predictive system. Under normal conditions, rigid, high-level beliefs exert a top-down constraint on perception, locking the mind into habitual patterns. Serotonin 2A agonism functionally relaxes the precision weighting of these predictive priors. The brain temporarily loses confidence in its established models of the self and the world. This relaxation results in a temporary state of enhanced neural entropy. Rigid belief structures dissolve and become highly available for revision. Precise instructive tone, 5-HT2A agonism creates a transient window of heightened flexibility, leaving the system open to contextual influence. If the pharmacological agent only opens the system, then the contextual influences operating inside that window determine the actual therapeutic outcome. This equation maps therapeutic change, or emergence, as a product of ground, reflection, and disruption. Delta squared represents disruption, the neural entropy introduced by the pharmacological input. Ground represents relational containment, while gamma represents reflection, the capacity to witness experience without becoming overwhelmed. Introducing high disruption into a system with low relational ground produces psychological fragmentation. In clinical settings, severe phenomenological experiences like ego dissolution are labeled as acute psychosis when the ground variable is absent. That exact same state of high disruption is recorded as a mystical breakthrough when ground and reflection are optimized. Elements like curated music, a strong therapeutic alliance, and structured preparation are not confounding variables to be controlled in a trial. They are the mechanisms of healing. If the process is the true mechanism, then non-pharmacological methods of altering consciousness should produce overlapping neural and therapeutic states. Functional MRI data confirms that sensory deprivation in flotation rest tanks and deep meditation practices disrupt default mode network connectivity in the same manner as psychedelics. Spontaneous mystical experiences share a nearly identical phenomenology. Historically, they have produced profound therapeutic identity change entirely without chemical induction. The anthropological record supports this. No documented traditional culture has ever administered psychedelics outside of an elaborate, containing ritual process. For 2 millennia, the Eleusinian mysteries served as the center of Western religious ceremony, utilizing a strict structure of preparation, revelation, and reintegration. The psychoactive substance consumed during these rites was embedded entirely within the ritual framework. It was never treated as a standalone cure. Traditional cultures treat the ritual container as a functional technology required for consciousness transformation, not as a cultural decoration. The modern pharmaceutical industry is heavily invested in developing psychoplastogens, compounds engineered to promote neuroplasticity while stripping away all hallucinogenic properties. The biological goal is to achieve structural neural changes, like rapid spinogenesis and neuridogenesis, without the regulatory complications of a subjective trip. This approach attempts to isolate the brain's physical hardware from its conscious software. Major mediational analyses contradict this premise. Data shows that subjective qualities, such as experiencing a mystical state or an emotional breakthrough, are the true predictors of long-term outcomes. During these subjective states, participants undergo an identity reorganization. They consciously realize they are no longer defined by their addiction. Structural neuroplasticity provides the biological capacity for change. The conscious reorganization of meaning dictates the direction of that change. Creating hardware change without a software update is a category error. Neuroplasticity is necessary, but without the subjective experience, it is insufficient. The current FDA pathway is rigidly designed to evaluate isolated molecules through phased clinical trials. Regulators evaluate psilocybin purely as a standalone compound. Evaluating a process-dependent treatment through a molecule-exclusive framework yields a clinically invalid assessment. It is equivalent to evaluating the efficacy of complex psychotherapy by only studying the physical couch the patient sits on. The current regulatory structure is blind to the variable, the container, that actually drives therapeutic efficacy in the data. In emerging state legal models like Oregon, a single legal psychedelic session currently costs between $1,500 and $3,200. Over 90% of that financial burden is attributable entirely to clinician time and process, not the pharmacological drug. The free market inherently recognizes that the human process and the time required to hold the container is the true mechanism of value. The intellectual property and the future of this field reside in optimizing the clinical protocol, the training, and the therapeutic relationship, rather than patenting synthetic molecules. The clinical evidence confirms that healing is a product of the human encounter. The chemical provides the access, but the meeting achieves the cure.